Chapter 7 Cellular Respiration. Here is a panda bear eating bamboo. But why do pandas and all other organisms need nutrients and energy? The only way for living things to do work is to have an energy source. Since life temporarily fights entropy and is non spontaneous, organisms need a constant influx of energy from chemical bonds in food. We now know that ATP powers our cells, but it can't be made without acquiring energy from the food that we eat. Food energy is found within chemical bonds of carbohydrates, lipids, and proteins. Ultimately, glucose is the molecule that houses energy needed to build ATP molecules. Sunlight powers life, but it can't be used in its raw form. Plants must convert thermal and light energy into chemical bonds of glucose, and the glucose bonds must be broken to transfer some of that energy into ATP. This process is called cellular respiration and is found in all living things on Earth. In cells, electrons are the most important source of chemical potential energy. In general, a molecule's potential energy is a function of its electron's configuration and position. The electrons in ATP have high potential energy because the four negative charges in its three phosphate groups repel each other. Hydrolysis of the bond between the two outermost phosphate groups results in the formation of adenosine diphosphate, also known as ADP, and inorganic phosphate. This is highly an exergonic reaction. The release of the phosphate group is transferred to a protein. The transfer of a phosphate group is called phosphorylation. So now that we know why and how ATP is used, where does it all begin? You see, glucose is a monosaccharide carbohydrate with 6 carbons, 6 oxygens, and 12 hydrogens. This excess source of hydrogens, as well as hydrocarbon bonds, is the source of energy needed to make ATP. Plants produce glucose from photosynthesis, while animals consume other organisms to get their glucose. The addition of oxygen gas helps to increase the amount of ATP made. During cellular respiration, the bonds holding glucose together are oxidized or broken down, releasing electrons and hydrogen ions. The energy released is then transferred between the beta and gamma phosphate groups on adenosine diphosphate and the inorganic phosphate. This condensation reaction results in water being made and ATP being formed. Unfortunately, there's more to making ATP than this. This is a huge metabolic pathway consisting of dozens of reactions split over 4 major processes. Please be patient. Don't consider changing your major as we go through this chapter. The only way to understand cellular respiration is to repeatedly look at it and find the patterns. Don't be intimidated by the molecular names. They're just names. Focus on the changes that happen between each chemical reaction, especially the changes that occur in the hydrogen ions and electrons. So why is hydrogen so important? For starters, there are 12 hydrogen atoms in a glucose molecule. Hydrogen has 1 proton in its nucleus, no neutrons, and 1 electron in its outer shell. Recall that when reading the periodic table, the atomic number refers to the number of protons. Protons and neutrons have mass, but electrons do not. Since hydrogen has a mass so close to 1, it virtually has no neutrons. Since hydrogen is neutral, the positively charged protons are canceled out by the negatively charged electrons. In metabolism, electrons are split from the atom, causing hydrogens to become positively charged. Each electron transfer from one molecule to another during a redox reaction is usually accompanied by a proton, also known as a hydrogen ion. Which brings me to my next point, a reduction-oxidation reaction, also known as a redox reaction. These are chemical reactions that involve electron transfer, and in this particular equation specifically, the electron transfers from hydrogens to other molecules. For this to work, you need a reducing agent. This is any molecule that will transfer an electron, thereby becoming oxidized. When we say the word oxidize, we mean that a molecule loses an electron. So in this case, the carbon group gives up a hydrogen. And since you know that hydrogens are made up of 1 proton, 1 electron, this becomes oxidized. Using the cell respiration equation below, Glucose is the reducing agent as it contains hydrogens which have electrons to be transferred. The oxidizing agent is a molecule that will receive an electron, therefore becoming reduced. Reduction is the gain of electrons. NAD plus is what we call an electron carrier. And what it'll do is it's going to receive that hydrogen ion and electrons from the reducing agent. Oxygen is the oxidation agent as it combined with the electrons provided by glucose in the cell respiration equation. So, reduction and oxidation. Reduction is the gain of an electron. Notice how NAD plus is reduced to NADH. Since oxygen, the oxidation agent, receives electrons from glucose, it becomes reduced to water. Oxidation is a loss of an electron. Notice that the carbon group is oxidized to carbon. Since glucose is the reducing agent transferring electrons to oxygen, glucose becomes oxidized to carbon dioxide. Oxidation and reduction events are always coupled, meaning that if one atom loses an electron, another has to gain it. Nothing disappears or reappears, it's just moved around. Electron donors are always paired with electron acceptors. Glucose is the electron donor and oxygen is the electron acceptor in cellular respiration. So a little bit more about electron carriers. How do metabolic reactions get done if positively charged hydrogens and electrons are free floating about? They're transported around via electron carriers. Electron carriers easily bind to one hydrogen and two electrons, causing NAD plus to turn into NADH. One electron binds to the NAD plus to become NAD, and the other electron binds to the hydrogen ion to become hydrogen. The result? NADH. Think of electron carriers as taxicabs or empty Ubers or Lyft cars, with vacant cars being NAD plus and people as electrons and hydrogen ions. When the car carries two electron passengers and one hydrogen ion passenger, it becomes an occupied car called NADH. When it empties the car at the destination, it goes back to being NAD plus. Therefore, it picks up more positive hydrogens and electrons. In aerobic cellular respiration, NADH takes hydrogen ions and electrons to the electron transport chain. Photosynthesis and cellular respiration use different electron carriers. FAD and FADH2 is only used in the citric acid cycle, which we will discuss later in this chapter. If positive hydrogens and electrons are being used to make ATP, where do they go when the job is completed? Well, they're picked up by electron acceptors. There are several electron acceptors throughout cellular respiration, but the final electron acceptor completes the process. Have you ever wondered why we breathe oxygen? I mean, seriously, you've been sucking this stuff in your lungs your entire life and you've never even thought to ask why? And maybe your answer is, well, we need it to live. Well, why? Why do we need it to live? Well, that's because oxygen is the final electron acceptor when performing aerobic cellular respiration. Oxygen is an energy hog. It loves electrons, making it electronegative. So these red dots are electrons. So being attracted to electrons that are used during cellular respiration, these electrons then bind to the oxygen, making the oxygen extra negative. And since the positive hydrogens are also attracted to these electrons, their combination with oxygen neutralizes the charges, creating water. So the whole point of cell respiration is to make ATP. Well, there's two ways in which this occurs in our cells. All ATP production is made by phosphorylation, which is the transfer of a phosphate group to ADP to make ATP. The cell does this in two ways. First is substrate-level phosphorylation, which usually occurs within a metabolic pathway in which an intermediate substrate carrying a phosphate interacts with an enzyme. The enzyme then transfers that phosphate to an ADP. This occurs during glycolysis and the citric acid cycle of cellular respiration. Oxidative phosphorylation occurs in the mitochondrion by harvesting hydrogen ions and electrons from glucose and using those hydrogen ions and electrons to help power a special enzyme called ATP synthase. This enzyme is what makes most of the ATP in your all cells. So before we get started with the process of cellular respiration, I want to introduce the 4 major chemical reactions that occur. If this table seems overwhelming, that's totally fine. Come back and review it after we go through each step. Right now, I'm going to summarize each. First of all, glycolysis, oxidation of pyruvate, and the citric acid cycle all function in the harvest of hydrogen ions and electrons off of glucose, so that they may be used to power the electron transport chain to make ATP. These 3 stages are collectively called the energy harvesting steps. Please note that some ATP is made in these steps, but only a small amount is made, and it's all by substrate-level phosphorylation. So let's break this down a little bit. So glycolysis, oxidation of pyruvate, and the citric acid cycle all are charged with stripping that original glucose molecule of all of these hydrogen ions and electrons. These guys are then picked up by electron carriers called NAD+ to become NADH. In glycolysis, 2 ATP is made by substrate-level phosphorylation. These are the important products and where they go. ETC stands for electron transport chain. This next part converts pyruvate, which is the end product of glycolysis, into a molecule called acetyl coenzyme A. So in English, basically, that allows the pyruvate to be converted into a form that can be sent to the mitochondrion, where these 2 steps take place. Again, take a look. It frees up more hydrogen ions and electrons, and that's the point. We're going to continue to strip as many of the hydrogen ions and electrons off of glucose as possible so that we can power that enzyme ATP synthase to build ATP for the cell. And that's exactly what the citric acid cycle does. It finishes that process of harvesting hydrogen ions and electrons. So anytime you see these guys, that means they've picked up all of those. They're transporting those hydrogen ions and electrons, and they always go to the ETC because that's where we're going to make ATP at a much larger scale. Of course, there's other byproducts. You'll notice CO2 gas gets expelled. That's normal. That is just the waste products of cellular respiration and the reason why we breathe out carbon dioxide. The electron transport chain takes all of the energy harvested, all the hydrogen ions and electrons harvested here, and this is where ATP is going to be made. And it's made by oxidative phosphorylation. Remember, phosphorylation is a transfer of a phosphate group to an ADP. And the reason why it's oxidative is because we are oxidizing glucose, in other words, freeing all the electrons and hydrogen ions out of glucose in order to allow this phosphorylation to take place. And in the end, oxygen is going to collect all those hydrogen ions and electrons once they've been used up. So where do they go? Oxygen grabs them, and they're going to use it to make water. So I want to at least kind of show you where we are in the cell. First, glycolysis takes place in the cytoplasm of the cell. Because of this, all cells, prokaryotes, eukaryotes, plants, animals, fungi, amoebas, zebras, all cell types undergo glycolysis. It's probably the oldest metabolic reaction on the planet, one of them. And so it doesn't require an organelle. Also, glycolysis doesn't require oxygen. And we're going to see later how we do cell respiration without oxygen. So if an organism undergoes anaerobic cellular respiration, this is about the only thing that they can do is glycolysis. Now, the next 3 steps do require the mitochondrion. Pyruvate oxidation takes place outside of the mitochondrion. The citric acid cycle takes place in
the mighty mitochondrion,
specifically the matrix of the mighty mitochondrion. Then the electron transport chain takes place in the inner membranes of membrane folds called the cristae. Note that the folding of these cristae increase the surface area of the membrane, allowing for more ATP production to take place. So all around here is where ATP production is going on. Now, since prokaryotes lack mitochondrion, the electron transport chain actually takes place on the cell membranes of the prokaryotic cell. And since the ETC takes place on the membranes of the mitochondrion, this kind of provides some evidence that the mitochondrion was at one time a prokaryotic cell. that was taken up by a larger eukaryotic cell. So evidence for the unity of life. In our next video, we're going to look at the first reaction, glycolysis.